I will be releasing your exam three grades later this evening. I am in the midst of wrapping up your final exam, um, and I'll talk about that a little bit and best ways to study for it. I elected to not do a practice exam, and I'll share kind of why in just a little while. But one thing that I wanted to do was I wanted to talk a little bit about DNA, and that was on our last lecture, but we're going to talk about it today. So much of what if you're a biology major, for example, you've probably taken or will be taking a molecular biology class. Um, and you know, that's a place where you would talk about DNA quite a bit. But if you are not a biology major, then you will not be taking that sort of class, or it's unlikely that you'd take that class. But I wanted to talk a little bit about nucleic acids and nucleotides, and give you some terminology that's important for you guys to know. And then I'll talk about how this fits into your final exam after a little while as well. So we're going to talk about nucleic acids, nucleotides, then we're going to pivot and we're going to go on to talk about the final exam and preparation for the final exam. So, first and foremost, when it comes to a DNA molecule, you've got a sugar. Um, and that sugar, more times, well, in the case of deoxyribonucleic acid and ribonucleic acid, so DNA and RNA, your sugar is ribose. Are there any nucleic acids that do not have ribose as the sugar? I'm sure that there are, but they're not, to my knowledge, involved in any sort of like earth-born organism. And anyway, that's a fascinating idea. Could you make a synthetic organism with synthetic or ribonucleotides that are not ribonucleotides, but instead would be like gluconucleotides? I don't know. But you could certainly make those molecules. Gluconucleotide, well, they're going to have a couple of things. Um, in both cases, we've got a sugar. That sugar is either ribose or deoxyribose. Um, and here is an example of a ribonucleotide. Here's an example of a deoxyribonucleotide. The difference between these at the sugar level is the placement of a hydroxide group at this carbon 2 prime. This is important. It's at 2 prime, not 2. Okay. Now, structurally, so I'll get into the numbering in just a little while. But it is at the 2 prime position as opposed to number 2, so that 2 prime is significant. Um, Now a nucleotide, so with a T, consists of a nitrogenous base, right here. A sugar, right down here. And at least one phosphate group. Okay, so when I was talking a moment ago about, well, could you have something other than ribose? Yeah, sure. Could you make a gluconucleotide? Nucleotide? Yeah, sure. All that you do is you take the sugar, replace the ribose with glucose. Um, but that's really just the kind of things that that would be at a cool interface of biology and chemistry and synthetic biology. At any rate, that would be kind of a cool thing. So that ribonucleotide, and deoxyribonucleotide, that's because of the sugar ribose. Now, nucleosides, that S, I'm going to erase some of this over here. Nucleosides are a little different because all that they have is the nitrogenous base here and the sugar. So the difference between a nucleoside and a nucleotide is phosphate. For some reason, I what always sticks in my mind is tide and phosphate. I don't know why. Yeah. So that sort of sticks in my head. I'm sure there's tons of other good. This PowerPoint is the last half of our most recent one from a couple couple days ago. So it's I think it's slides like 16 through 25. We didn't get to them on the last time that I got that as a private message. So I was just responding to that. So this is on Canvas, but it's the last half of the last one. Sorry about that. I hope nobody's losing their minds about that. Because yeah, that yeah. Okay. So nucleotides. We've talked about these quite a bit. Specifically, we've talked about ATP. Now, one thing that's worth noting, whenever you talk about ATP versus DATP, well, there's a difference. DATP is for DNA, whereas ATP would be a ribonucleoside. Now, one thing that's worth noting is this terminology. We talked about nucleotide versus a nucleoside. Look at this right here. Ribonucleoside 5' phosphate. If I were to take, what's another word for a nucleoside 5' phosphate? I'll give you a hint. It starts with ribonucleo. Yeah. A ribonucleotide. Okay, so I know that was a little bit of a leading question, like, hey guys, this is my fantastic question that I have in my mind the exact answer that I'm looking for. I hope that made some sense for y'all. Um, why did they not just say that? I think that it all boils down to that's a great question. What it's going to boil down to is a I think nucleotide is a good word to have to, talk about a class of compounds. But then when you say ribonucleoside or nucleoside 5' phosphate, it gives you a little bit more flexibility to provide a little bit more detail. So I will quite commonly refer to ATP as, or ATP is an example of a nucleoside, oside, triphosphate. I could also say that it is phosphate. It's also a nucleotide. Both are acceptable. Um, I think I have a tendency to say nucleoside triphosphate just because then it gives me the kind of assurance or that certainty. Yes, there's three phosphates there. So worth noting. So at this point in time, if you're thinking, okay, what should I be thinking about with respect to the exam, and what sort of questions should I anticipate? Definitions, nucleoside versus a nucleotide. That's a great question to be able to answer. If I showed you, for example, this molecule right here, and I said, okay, this has which of the following? Nitrogenous base, a sugar, a phosphate, a carboxylate. Well, that would be a question about all the different functional groups. But then I could also ask just the general question, what is the best definition for this molecule that's shown here? A nucleoside, a nucleotide, a amino acid. Just those are good things to be able to answer regarding this. Okay, next up, the nitrogenous bases that we find in a nucleoside triphosphate, or heck, a nucleoside for that matter, are categorized as either a pyrimidine or a purine. Between let's see, the 46 of us, whenever I was an undergrad, yeah, 20 years ago, it happens fast. Um, so my genetics professor was Dr. Forrestner. And every time I mention that, people are like, what? Yeah, I'm Bobcat through and through. And Dr. Forrestner was my genetics professor, and I think he did a postdoc, or maybe he did his PhD at Texas A&M. I think it was a postdoc. And the he gave us a mnemonic and he said, well, purine. Okay, purines are a two-ring structure. The nitrogenous bases for purines are adenines and guanines, or adenine and guanine are your nitrogenous bases. And the mnemonic was ags are pure. And so just a little one to remember. And that's really just my way of bringing up that Dr. Forrester was my professor. So anytime I think, man, Joel, you're getting old. Well, Dr. Forrester's older. So there we go. Um, at any rate, he was my genetics professor. Um, so the other nitrogenous bases are pyrimidines. Pyrimidines are going to be, if you're talking about a DNA molecule, cytosine, thymine. If you're talking about RNA, you're going to have cytosine or uracil. Um, it's good to know, and the single letter codes for these are C, T, U, A, G. Now, this is all pretty basic DNA stuff. Um, this is kind of a next level sort of consideration. We could have um, these nucleotides display in a couple of different ways. All of these molecules here, are they all nucleotides? And let's think about that. We need a nitrogenous base, we need a sugar, and we need a phosphate. Okay, adenosine 5' monophosphate. Adenine is our nitrogenous base, a ribose sugar, and last but not least, a phosphate. So that one's good. We move over here to adenosine 2' monophosphate. It's good. Down to the lower left, adenosine 3' monophosphate. And then last but not least, adenosine 2', 3' cyclic monophosphate. Yeah, put it short, these are all nucleotides. Um, with that in mind, this one in the lower right-hand corner is an example of a cyclic adenosine monophosphate, or CAMP. Sometimes you learn about CAMP as a secondary messenger, so being involved in signaling, like a cell perceives caffeine or nicotine or something like that. Well, that nicotine isn't going to cross the membrane, but rather it's going to stimulate some sort of response. You're going to activate the production of adenosine mono or cyclic adenosine monophosphate. That's going to lead to basically triggering on a whole bunch of different enzymes to stimulate either gene expression or maybe they'll activate beta oxidation or something like that. They'll change metabolism as well as they'll change genes and protein production. So these are all monophosphates. These are all nucleotides. Now, at this point in time, when we look back at these, what I'm going to do is I'm going to say you should expect to be able to identify based on a structure your um, nitrogenous bases. And what I mean by that is if I give you a structure like this, if I give you the general structure of a pyrimidine, I can say, well, is this cytosine, thymine, purine, or pyrimidine? Give you those different answer options. And I want you to be able to say, that's a pyrimidine. I want you to also be able to identify something based on its structure and say, well, this is cytosine. Now, the way that I do that and giving you kind of the boundaries of I'm not going to be asking you to draw these, but instead identify them, is I'm going to look at all of these. I'm going to start down here with my pyrimidines, and I'm going to say, okay, what do I need to recognize about my purines or pyrimidines? I'm sorry. I always think about these as the faces of a clock. And what I'm going to do is I'm going to say cytosine at 12 o'clock has an H2. This is my 12 o'clock position. I'm looking at the face of an analog clock. That gives me my way of differentiating between cytosine, thymine, and uracil. I also know that in order to go through that process, I know that a pyrimidine is a single ring structure. Now, thymine, okay, well, you know what I'm going to do is I'm going to kind of make it easy on myself. And I'm going to say Thymine, represented by a single letter T, at 2 o'clock or 2 has CH3. By knowing those two, do I need to spend any time thinking about the structure of uracil? No. Process of elimination. I'm going to see this structure and I'm going to be like, okay, well, that's uracil because I know that it does not at the 12 o'clock position have a nitrogen. And at the 2 o'clock position, well, it doesn't have a methyl group. So by process of elimination, this is uracil. Done. Now, for adenine and guanine, well, adenine and guanine, I'm going to kind of do the same thing. One, I'm going to know that these are purines because, well, they're two ring structures or bicyclic structures. And what I'm going to do is I'm going to think 12 o'clock, I've got NH2 for 4A. Then guanine, that's simply put my other bicyclic structure. Okay, so those are my, so at this point in time, thinking about what do I need to know for this exam? Nucleoside versus a nucleotide. Be able to differentiate between them. In addition to that, numbering of the carbons of the sugar ring, which we're going to get into a little bit more, but specifically differentiating between a deoxyribonucleotide and a ribonucleotide by looking at the 2' carbon. This is the sort of thing that is it seems so trivial and so simple, but people will get this question incorrect. If I give you these two structures and I say, okay, this one is A, ribo, dot, 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 B, deoxyribo, and then C, let's say glycine. A surprising number of people will select deoxyribo. Why? Chances are, if they're anything like a younger Joel Gray who took Dr. Forrestner's class years and years ago, chances are they are saying, ooh, deoxy, ooh, oxy, that has an oxygen in it. Perfect. Done. Okay. Why did I do that? I have no clue, but that's just a silly, foolish thing where I somehow was like, why didn't I look at the two letters that preceded oxy? I don't know, but here we are. So I want you to know the numbers. I want you to be able to identify these different molecules. I want you to know the difference between a nucleotide and a nucleoside, be able to identify a nitrogenous base by its structure. Um, in terms of, this is another thing that I just want to highlight. Our numbering of our phosphates are alpha, beta, and gamma. Gamma is the phosphate that comes off in a kinase reaction or in a transferase reaction where a phosphate group is transferred away. Am I going to ask that specific question? No, I'm not. But if you take metabolism next semester and you talk about a gamma phosphate, that's the reference point there. In the event that you hear something taking place above me or you hear lots of noise, I don't know what that is. I think that it probably has to do with my kids, my dogs, and dishes. So here we are. We got a couple monophosphates, and there we go. Those are the big things. Now, the when it comes to this numbering, I talked about this a little bit, this numbering 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. We use that numbering because the nitrogenous bases, they all have their own non-primed numbers. So this allows us to identify specific location on the sugar of a ribonucleotide or a deoxyribonucleotide without having to change the numbers. What I mean by that is how many atoms are there in a purine? There's nine within the, sorry, there's nine atoms that make up the two rings. Okay, so if we were to look at a purine that connected to ribose. Well, this nitrogen is atom number 9 in that ring. So if we were talking about connecting that to our ring structure here, add to the chat what number atom or what would the number be that I put right there if I was kind of going in a sequence and I didn't have the prime numbering scheme. Probably 10. Yeah. Okay. Now let's think about this right here. Pyrimidine. Oh man, that gets our priority right there, number one. So then we got the same sort of thing. We've got 6 atoms up here. We'd have to come up with a way to number this atom number 7. So you're talking about the exact same position on two very similar molecules, and their numbers are 7 and 10 respectively. That would, create a kind of confusing environment. So we bypass that altogether and we just go with the prime numbering scheme of 1 prime, 2 prime, 3 prime, 4 prime, 5 prime for the atoms within the sugar ring. Okay, so in addition to um, nitrogenous bases and nucleotides and nucleosides being a part of transmitting genetic information and being the carriers of genetic information, they're also involved in a number of other things. Now, have we talked about any of these molecules? This one, this one, this one. Yeah, we talked about those. NAD plus FADH or FAD, those were great molecules and compounds that we talked about in the context of the citric acid cycle and oxidative phosphorylation. What were those things really good at? Being electron carriers. So, what is a part of those two molecules? Well, NAD plus is also known as nicotinamide adenine dinucleotide. That is a compound that, well, it has a couple of nucleotides within it. And, well, that's just kind of the how dynamic nucleotides can possibly be. Now, when it comes to, so this is just kind of trivial knowledge for right here. When it comes to bonding of DNA molecules and RNA molecules for that matter, the type of bond that connects one sugar to another sugar is known as a phosphodiester linkage. This phosphodiester linkage is the bond that connects these polymers. These polymers are linked in a 3' to 5' direction. The ribonucleic acids and deoxyribonucleic acids, also known as RNA and DNA respectively, are held together by these phosphodiester linkages. The sequence is always read in a 5' to 3' direction. So we have our 5' end right here, and we read it in this direction. So we read it as, here's our sequence, A, T, G, so on. This is analogous to the sequence that we use for reading proteins and termini to C termini. Now, lots of this right here is probably not particularly new. But one thing that I want you to think about is when we look at this right here, let's imagine that you didn't have this part of the molecule. If I showed you only this snapshot of my molecule, would you be able to identify this as a DNA or an RNA molecule? We got an RNA. You're absolutely right, Megan, because we've got that 2' OH group. So be kind of vigilant of looking at that as a piece of, or as a context clue. Now, when you read the sequence of a DNA molecule, there's a couple of things that are worth noting. This right here is essentially the same as this right here. This model shown on the left, or the sequence shown here on the left, is a little bit redundant and a little bit excessive because what this is demonstrating, these lowercase p's just indicate where this molecule has a phosphate. Now, what I want you to do is looking at this molecule right here, or looking at this sequence right here, I want you to think about this and read this as this indicates our molecule is phosphorylated on the 5 prime end. Based on this sequence that I've got a big circle around and I just put two stars around, is this molecule phosphorylated on the 3 prime end? Black boxes. I got black boxes. Okay, so I'm going to go ahead and I'll try this and see this happen. Uh, what about now? Oh yeah, I see. That's so weird. That is too bizarre. Okay. I see two black boxes. Now I see one massive one. Let's see. You know what? Let's see if I can do it this way. I'm going to try to do it not as a full screen and see if that has any impact. Oh, that's so bananas. I see it in my like um, yeah, that's kind of weird and it's borderline eerie. Oh, you know what? Did one of them just move? Did one of them just disappear? Now I can't look at the chat. Okay. So whenever I have the chat up, that is one of the boxes. The other box is like my menu item. It is 100% bananas. Okay, so what I'm going to do is I'm going to I think I can go back to full screen because that makes it easier to use my stylus. No, wait, maybe. But I've got some limited, Kevin, I appreciate that. I too. I like banana bread. My wife made banana bread muffins. Um, what I'm going to do is I'm going to close my chat and I'll pop back in periodically to look at the chat. So, okay. And then I'll just have to move my bar around, move this around every so often. Let's see, show me. No, hide meeting. Thanks for nothing, Zoom, except for the technology to connect people around the world. Okay. So, so what we have here is this. It's my Marge Simpson. Oh, me. This molecule right here is not phosphorylated on the 3 prime end. This lowercase p right here indicates that it's phosphorylated on the 5 prime end. So if I were to show you something like this, PACGTCP, this is phosphorylated on the 5 prime end and the 3 prime end. And you might be thinking, how do we know? Well, the what we know is that at the ends of our molecule, we have a 5 prime carbon and a 3 prime carbon. If we indicate this lowercase p, or if we show that lowercase p, that indicates that rather than an OH here, we have an O, P, O, O, O. So then if I were to show you something like this, A, G, G, C, P, this molecule is 3 prime only. And then if I were to say A, G, A, This is not on the 5 prime nor the 3 prime end. Now, the significance of this is that if a molecule is phosphorylated on the 3 prime and 5 prime end, or on neither ends, well, that's particularly significant for how that molecule can be used for either PCR, so the polymerase chain reaction, but you can basically think about it as the 5 prime or 3 prime phosphorylation is going to impact the biotechnology uses for that molecule. Our overall structure for our DNA molecule is the secondary, or sorry, is the double helix. We've got a couple of people to thank for that. We've got Maurice Wilkins. We've got Rosalind Franklin. Then we've got sorry, we've Watson and Crick. These were all seminal and very important figures in the elucidation and theorization of the DNA double helix. And it's a sugar phosphate backbone. And in the center of our molecule, that's where our bases are going to hydrogen bond with one another. The backbone is considered anti-parallel, and there are major and minor grooves that make up the structure. What I want you to take away from this is this right here and this right here. These phosphodiester linkage make up that sugar phosphate backbone on the outside. The hydrogen bonds are in the inside of the molecule. After that, I want you to recognize the numbers of bonds. If we've got guanine here and cytosine here, these two molecules can make one, two, three hydrogen bonds between them. G and C pair up, A and T pair up. They're complements of one another. So anytime you see a T, you can predict that on the other strand of that molecule, you've got a T. Yeah, where you have a T, you have an A. Where you have an A, you have a T. Where you have a C, you have a G. Where you have a G, you have a C. They're going to complement one another. Now, this is commonly known as Chargaff's rule or Chargaff's rule. This is also known as Watson-Crick base pairing. Chargaff's rule is the number of A's that you have is equal to the numbers of P that you have. The numbers of G's is equal to the numbers of C's. There's some simple math that you can put together. If I tell you that a DNA molecule is 20% C, well, as it turns out, I've told you everything about the composition of that molecule because Chargaff's rule says if it's 20% C, then it is 20% G. Those two combine to give you 40%, leaving behind 60%, which means these two things that are equal to one another, A and T, it's 30% A, or yeah, T, and 30% A. So that's our total breakdown of this. Now, the significance of this is, well, when we start talking about really large molecules, if you have something that is, let's talk about two different molecules, molecule A and molecule B. Let's say they're the exact same length, 1,000 base pairs. You can come up with an idea of how strong these molecules are based on their ATGC content. If you've got a molecule that's 1,000 base pairs and it is, let's say, 20% C, and then you've got another molecule, or I'm looking at molecule B now, and I'm going to say that it is let's go with 45% T. Which one is stronger? Well, to think about that, or to answer that sort of question, which I'm guessing someone's going to be adding to the chat very shortly, um, and I'm happy that, oh, damn it, stupid thing. All my text just disappeared. Okay, I can do this again. Molecule A, molecule B, 1,000 base pairs. 1,000 base pairs. I'm happy that I got myself on video recorded swearing. Okay, molecule A is 20% A. Molecule B is, I said 20% C. 45% A. There we go. We know that this is 20% G. We know that this, and then that means that the remainder of this is 30% A, 30% T. Our other molecule B is 45% T, 5% G, 5% C. We know all that. Okay, so which one's stronger? Well, ultimately, it boils down to looking at these individual interactions. We've got three H bonds between G and C, two H bonds between A and T. So essentially, what that means is if you have something that's a lot of GC content or a high GC content, that is stronger than something that's low GC content. So stronger is determined by GC content. Okay, so that gives us A is our correct answer. And yes, the chat is back, so that's why a portion of the screen's blocked out. Okay, now nucleic acid manipulations. If you took genetics and the lab is anything like it was whenever I took it with, again, Dr. Forrester was my lecture professor, then you would have done, we had our TA did PCR for us and we ran a DNA gel. It's a way of separating pieces of separating nucleic acids from one another based on their size. The polymerase chain reaction, also known as PCR, something developed by oh, something that was developed by Kerry Mullis is credited with it, and he's a colorful character who's now deceased, but he was a colorful character, and basically it utilized a series of steps to take one double-stranded piece of DNA to make thousands of them. This is the sort of thing that we kind of take it for granted because it's so simple and so fast and so easy to do now. Wasn't always the case, and this is kind of the like this moved biomedical research ahead by, I don't know, I can't think of a specific number, so I'll just say a lot. This is tremendous. This is a great discovery, and being able to utilize this and harness this. One thing that I always think about is my graduate advisor told me, back in the like 1970s, if you were able to sequence a gene, that would probably earn a PhD. And the reason for that was the tools that you had at your disposal. It would take the better part of 6 or 7 years to get a definitive sequence on a gene. Now, because of PCR, we can sequence DNA in a different way. And basically, we can sequence it. I mean, if you don't have chances are any average gene you're going to have the sequence of within 48 hours after you know, isolated it, perhaps faster than that. Depends on access. But um, this is like years down to a matter of days. So it's pretty awesome. Now, what this consists of is three major steps: denaturation, hybridization, and polymerization. These steps take place at different temperatures, and that's what I want you to know. I want you to know these temperatures and temperature ranges. In order to do PCR, you have to have DNA primers. You have to have a heat stable DNA polymerase. That's the biggest thing right there. A heat stable DNA polymerase, something that can sustain a DNA polymerase, just like what we've got in our bodies, the enzyme that makes DNA polymers. We have to have one that can endure heating up to around 98 degrees, which is what you do for the denaturation. Which, when I was an undergrad and we had mentioned 98 degrees, you'd think of the pop band, but they're long since gone. I think they're all still alive, but no longer popular. Then you have this hybridization step. That takes place at about 55 degrees. And of course, these temperatures are a little bit variable. Then the polymerization, where you're actually synthesizing the piece of DNA, that's going to take place at about 72 degrees Celsius. So these are all degrees Celsius. If you do PCR or have done PCR, your temperatures vary a little bit, anywhere from probably 95 to 99 for denaturation. The hybridization and polymerization might vary a little bit more depending on the gene that you're trying to amplify. Now, to visualize these steps, right here, we're separating our strands by heating them up. So we're going from this to this and this. That's the separation. Then what we've got is the hybridization. This is a DNA primer and this is a DNA primer. They complement regions of the piece of DNA that we're trying to amplify. So this 98 degrees, 55 degrees. So we heat it up, then we cool it down. Then at 72 degrees, we have this polymerization take place. And then what happens after that? Well, we start it all over again. We repeat that same cycle. Heat, cool, heat up a little bit more. Heat, cool, heat up a little more. Heat, cool, a little bit more. So we go from one piece of DNA to two after one round, to four after two rounds, to eight after three rounds, 16, 32, 64. I, I, I can go beyond that. Check it out. But I'm not gonna really try to boast too much. Um, Applications of PCR, they are vast and many. Whenever I was an undergraduate, and we talk about DNA, sorry, when we talk about PCR, it was basically like, hey, do you remember the O.J. Simpson murder trial? And that was pretty much it. That was before we lived through a global pandemic where, hey, if you want to do this, you got to have a PCR test, a negative PCR test. Um, but that was, yeah, 20 something years ago. Okay. Does anyone have any questions about DNA or nucleic acids? I can open up my chat now and not obstruct half the screen or a portion of the screen. What is there? There we go. Right. Okay. Um, Now, what I'm going to do, and you guys can have questions if you want. You know what? I can do that, Brianna, and I'm going to do that, but in a Word document. So, let's see if this obscure. Yep, it does. That's so bananas. Okay. Okay. So, um, lowercase p, a, p, that is that notation right there indicates you have a nitrogenous base of an adenine, a ribose, or possibly a deoxyribose. Well, actually, in this case, it would indicate a ribose, and that this molecule is phosphorylated at the 3' and 5' end. Now, I can zoom. Let's go through 5. I can highlight, and then go through. Um, on the left-hand side, I'm going to highlight this with yellow. That indicates I've got a 5' phosphorylation. I'm just choosing these colors so that I give you some sort of illustration. Then we've got red over here. That's our 3' phosphorylation. So what you were asking about was what about P A P U P C, and then we'll go with one more lowercase p. Okay, this, which I'm going to underline, that, which I'm going to underline, but then I'm going to add this to my dictionary. The two underlined phosphates, I mentioned that we kind of take those for granted due to the fact that, well, how else? Like those indicate those are illustrating the phosphodiester bonds. Those phosphodiester bonds, um, those phosphodiester, um, those kind of phosphodiester bonds are more or less taken for granted because of the fact that there's no way for the A and the U and the U and the C to connect to one another and be a nucleic acid without this P. That's why it's pretty common that you won't see this notation, but instead you'll see this PAUCP. Because what's significant from like what's that molecule look like is, well, this indicates it's phosphorylated on the 3' end and the 5' end. I got a private message. Can I upload the summaries and study notes for the 16th and the 14th? I will do my best to do that. I'm going to make a note of that. Note 4375, 414, and 416. I will try to take care of that this evening. So one of the questions that you might field for this sort of thing would be like, um, ACG lowercase p, or heck, I mean, this would be this that right there is your boilerplate, your basic sort of question, or your basic sort of prompt, the one that I've got highlighted right now, not the one that's highlighted in pink. Okay, that one that I have highlighted in gray. This is phosphorylated on the 5' or the 3' end. Well, you got to think about 5' is on the left-hand side, 3' is on the left hand or on the right-hand side. So this is phosphorylated on the 3' end. This right here would be, well, it's phosphorylated on the 5' and the 3' end. I could also say this right here, PPACGP, this would be, well, it's doubly phosphorylated on the 5' end, triply phosphorylated. That's just kind of a wrinkle to that sort of question. Chances are you're going to get a question that asks about this and gives you this example or this example. And I'll give you a little bit of a spoiler. If I gave you P, A, C, G, P, chances are slim I'm going to give you that because is this only phosphorylated the 5' end, the 3' end, both, or neither? Well, you're going to look at that and be like, well, got it down to both or neither. Process of elimination, it's probably both. Okay. So the P's in between are just understood. Yes, absolutely right. Okay, so now what I'd like to do, if anyone's got any questions, whether it's about DNA, the final exam, or anything like that, please don't hesitate to interrupt me. Either send me a direct message or send me or you can unmute yourself, send an overall message. I do have, uh, let's see, final exam. I do have ADHD, so chances are if I'm in the middle of an explanation, I am going to stop and try to answer the question that pops up in the chat, and I apologize in advance for that. So the first thing is I'm going to take the exam schedule for the university and add it to the chat. Registrar, Texas State, persistent links, blah, 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 blah. And then I'm going to share my screen so that we are all on the same page here. I believe on our Canvas page, I've got the exam listed as being, I I got a question about nucleotides, and DNA on the file. I'm going to get into that in just a little bit. We've got a Monday, Wednesday class from 6:30 to 7:50, 7:21, so good. This exam will start at 8 p.m. and go until 10:30, and it is this Wednesday, 30th in the year 2025. Okay, so I'm going to go back to my document. I say exam. Oh yeah, go up to, let's go to, I think 25 is a good. There we go. Exam, Wednesday, 8 p.m. to 10:30 p.m. And I will add in parentheses here, um, April 30th. Then I think a fair question is the breakdown of the exam. Or the exam is going to be 85 questions, but it's going to be graded out of 80. So what that means for you guys is if you get 80 questions right, ooh, ya, ka, sha. That's a 100%. If you get, and if you don't know what Booyakasha is, that's from Ali G. Do we have to take the exam during the time or can start earlier? I'm going to get to that in just a moment. I'm going to have a little bit of a buffer there, so you will be able to start a little bit earlier. If you get 85 correct out of 80, then that is a 106.3%. So 5 questions essentially are bonus questions. They are, it's not like question number 14 or anything like that. You just have to, go with the exam that there is, and that's how I'm going to score it. Next up, you'll have 2.5 hours to do the exam, and it is on Proctorio. I will start the exam window at 7 p.m. I think that's a good amount of time. Um, so if you want to start then, if you start then, your exam block will close at 9:30 p.m. I will be on Zoom starting at 8 p.m. and we'll stay until 10:30 p.m. Zoom is not mandatory, but I will be available if you have questions. If you want to just join in and sit and watch or sit and, have me be there, that's fine. My camera and my audio will be off, but I'll be at my computer if I step away. If you ask a question and you don't get a response, um, I don't think that I will drift very far from my computer, so probably within 3 minutes or so, I'll get back to you. Hopefully, it will not be anything more than that. Now the next thing is the breakdown of the exam. Where are those 85 questions coming from? What I'm going to do is I'm going to say about 10 to, around 10 questions are DNA. And I'm going to say 8 to 10. I think that I gave you some good ideas of what to anticipate. We got numbering, we got identifying. Um, those are good things that you should kind of expect and anticipate. 8 to 10 questions on DNA. And so Is the definition of nucleoside or nucleotide going to be on there? Yeah, 1000%. Um, so be ready and able to do that. Identifying a nitrogenous base, yeah, at least one will be on there, possibly two. Um, Identifying bonds, yep. Identifying strength of like comparison of two molecules, which one is stronger, something that has this percent A, this percent G, this percent T. Yeah, those are very good and what I think are kind of easy questions. Um, but those are good places to go in terms of kind of breaking down those 10 questions. I got a private message question of, sorry if I missed it. We can still use a note card, right? Well, here's my flair for the dramatic. No, you cannot use a note card. Instead, you can use one 8, no, 8.5 by 11 piece of paper. So essentially, that's like four note cards. Yeah, sorry, I couldn't resist. And I feel like my comedic delivery, I didn't sacrifice anything there. So um and front and back is good to go. I feel like if someone from the 1800s read this transcript, they'd be like, "What the heck does that mean? Front and back is good to go." Speak in words, Joel. Let me rephrase that then. You can write on both the front and back of the paper. Um, Handwritten or typed, both are fine. I did have a question from one of my students earlier today, and they said, does it have to be printed? And we had to, it went back and forth for a little bit. Um, you do not have to have something printed, like it doesn't have to be coming from a printer. What I mean, or what I think is a better way of thinking about it, is it has to be a hard copy. So I don't want anyone to be like, oh, well, I use here's this 8.5 by 11 on my iPad. I use that. That's not a viable option. Um, Standard notebook paper is a good way to go, but at the same time, if you still have your note cards from your previous exams and you want to take pictures of those and throw those, shrink them down and put them onto this piece of paper, go for it. I have no qualms with that. Um, get creative, do what you want, but at the same time, be judicious with your time. Um, I wouldn't spend like the next 36 hours figuring out the way to be most efficient with that paper as possible. I'd spend some time I put amino acid structures on there, titration curve, PI calculation, those sorts of things. Those things that are like high value things, put those on there so I know that I'm getting them. Um, and then I would not, yeah, then I'd go back and just study, use practice. Like, can I still answer questions? Okay, so that brings us to roughly, I'll say 75 to 77 questions. Where are they coming from? And I mentioned, oh, well, I'm not doing a practice exam, even though I previously said that I was going to. And the reason that I did that was um, I kind of ran out of energy for one. I'll be honest with you. Run out of energy. And I don't even have to take well, I don't have to take an exam. You guys do. So please do your best to hang in there. Um, but another reason that I'm going to lean on a little bit more is um, the best way to study for this exam. I like to think that honesty is the best policy, and sharing with you that I ran out of energy, I think is, well, it is honest. Um, but this is your opportunity to be like, man, doing my evaluation, Dr. Gray, he's lazy. Best way to study for this exam is old practice exams and old exams, the ones that you've taken this semester. The reason for that is if you haven't been tested on something, it's not on the exam. Let me rephrase that. It's not on the final exam. If there's something that over the course of the semester I maybe said like, oh, this is super important, but I never ended up asking you a question about it on any of the exams, whether it's a practice exam or an actual exam, um, I'm not going to ask you about an exam now. I believe you can. My understanding was that you can access the previous exams. Number three is not available if they, um, but you're asking me this question, so that makes me conclude no, not the case. So I will look back at that. Um, so okay, I'll look into resolving that. Um, so no matter how, if you were over the course of the semester, like, oh, reading between the lines, he's going to ask us a question about this, but I didn't. That's not on the table. Will there be some repeated questions? Possibly. Um, some questions may be repeated. Yes, the third exam is going to be opened whenever I release the grades, which should be later this evening. So I will do my best to make sure that you have your old exams later today or later tonight. Um, but it's possible some questions are going to be repeated. Um, what I would do if I were you when it comes to like studying for exams is I would look at the questions that you've answered. Did you get them right? Why did you get them right? Did you get them right because you kind of lucked out? Um, or you just flipped a coin and you ended up with the right one? That's a great question. And quizzes, no, I'm going to be primarily focusing on the practice exams and actual exams. So I will give you that kind of range. If it was asked on a practice exam or an actual exam, that is within the realm of content that can be pulled for um, the actual final. But quizzes, I like to, there were some things that were asked on there that I didn't ask on a midterm. I'm not going to be digging back into those though. So are the 75? Yes. So basically that's going to be 20, it'd be like it's going to be pretty even. E1 would be, let's say it's 23 questions. E2 would be 24. I don't know how to math right now. C3 is 26, 24, 25. Yeah, 25. Yeah, we'll do 25, 26, and then E3, 26, Q, just as a hypothetical example. Um, I'm aiming for them to be evenly split. And then yeah, that's what I got there. Um, the last thing that I wanted to say was basically the way that I do grades. This is getting a little bit ahead of ourselves. Sorry, this is getting a little bit ahead of ourselves. A, B, C, D, F. An 89.5 and above will absolutely be an A. 79.5 to below 89.5 is a B. A 69.5 to below a 79.5 is absolutely a C. A 59.5 to below a 69.5 is a D, and anything below 59.5 is a failing grade, is an F. Now, okay, it seems like some folks can see exam one and two. Three hasn't been released, so but I will look into all of them. So, um, what I look for when it comes to grades is I will take everyone's grades honest to the lowest. And in a class this large, there's always going to be one person who scores like, they end up with a 100 or a 102 or something like that for the semester. Those people rarely are concerned with, oh, did I get a 99.7 or a 99.9? There are those people who do ask me that question. Um, but the people who are most interested in grades are the ones who are right at around the breaks. And what I do is I look for natural breaks, or what I've always termed natural breaks. I'm not sure if I heard that somewhere or what, but what I'm looking for is I'm going to line up the grades, and let's say that we've got someone who had an 89.51, another person who got an 89.49, 89.48, 89.46, and then last but not least, and this is from right around that range, these are students' grades. Let's say the next highest grade was an 88.62. Whenever I look at these grades, I'm not a hard line objective person. And so I see this and I think, okay, that person who got the 89.51, that is an A. Done. Easy. 89.49. Well, I did say 89.50, but whenever I think about how these two students performed over the course of the semester, did the person who got the 89.51, you know, I feel like it's almost just a flip of the coin that they ended up with a higher grade than the other one. And so what I do with that is I take all of these grades, and those are all A's, because the difference between these four grades, well, the 0.51 and 0.46, that's a decent difference, but the 0.51 to the 0.49 to the 0.48 to the 0.46, those are all so similar to me that I just look at them, I'm like, that's effectively the same grade. What that means is my highest B is going to be an 88.62, and that is going to basically replace this right here. And that's just a matter of I haven't really changed anything. I've just redefined, or sorry, let me rephrase that. Um, I haven't lowered the B's, but instead, well, the A's dip down a little bit more. And then the highest B happens to be here. The highest B happens to be lower than this. Um, some people want to call this a curve. You're welcome to. I'm fine with that. I don't have any qualms with that. I don't look at individual exams and say, oh, I changed, or I curved exam two, I curved exam three. I don't do any of that. I look at the overall grade because if somebody makes a 97 in a class for the semester and on exam one they got a 100 and on exam three they got a 93, doesn't matter. All that matters is the grade in the transcript. Nobody's going to see that you got a 93 on an exam. I don't mean to be pessimistic or nihilistic or negative in general. It's just the fact of the matter. So that's what I look for. To be completely honest with you, the A B range doesn't change very much usually. It's usually about an 89.5. The BC range, that changes a little bit more. What I see the most variation in is the CD range. So this might drop down to like 68 or 67 and a half. It varies from class to class, but the CD range varies a little bit more than the AB and the BC. Um, but that's what I do because yeah, Brianna, I see you, I hear you. The point oh 0.06, it always just kind of feels silly to me. But that's what I got. Um, does anyone have any questions for me? And you can email me questions. I will get back to you as soon as I can. Um, but if something comes up with the exam, like, hey, could I take it earlier? Yeah, I can sort something out. Yes, I did say the note card can be double-sided. In terms of the final replaces previous exams, right? Yes, it can, provided the final is higher. Um, if you'd like, I can share, put this little table together for one of my classes earlier today. I'll share it and I'll show it on the screen. I can't remember the exact percentage of each grade's value, but let me show you this table. I have one question. How does it feel to be the world's best professor? I'd love to know. I emailed you if you please take a look. Was it today? If it was yesterday or before today, sorry, please double tap on that. Um, and I will look at it. Let's see, document 3 is what I'm looking for. Okay, so okay, I will take a look at it later today. Um, I'm flattered that somebody thinks that I'm the world's best professor. That's very thoughtful. Thank you very much. Um, I'm glad to see that uh, that cheat sheet gives you some relief because that's what I'd like it to do. Okay, so here's a situation. Can we use our old index cards? So we'll have a total of 4. You're welcome to reuse them. What I think I would do is you know, I would transcribe that information onto a piece of paper just so it's a little bit more visible, a little easier to read. But you're welcome to do whatever you'd like there. So yeah, you can reuse them. Um, Now, okay, so here's what we've got. We had 3 midterm exams and we got a final. And this is a situation where exam number 1, person got a 95, 2, they got a 93, they got a 0. They missed it. And then they come in and they take the final, and let's say that they get for this case, let's say a 93. I can't remember the percentages. I think your class is 15% for the weight of each one of your exams. Um, but what I'm going to do is I'm going to copy, or you'll see kind of a category, or you won't see the category, but I assure you it's there. Category module 1, category for module 2, category for module 3, and then last but not least, the final. The final is worth 15% of your grade, so it is a requirement in the sense that if you want to earn 15% of your grade, you should take the final. If you're fine not taking that, then the highest grade you can get is basically an 85. What I'm going to do is I'm going to apply a rule so that each one of these categories, each one of these columns will get it will get a rule where the 2 lowest grades are going to be dropped. So the 90 and the 93, by comparison, are lower and therefore will be dropped. Module 2, we've got the 0 and the 93, or sorry, the 0 and the 90 will be dropped. Now, for module 3, there's only 2 grades, so only the lowest one will be dropped, and that in this case would be a 0. So in this case, um, this person took their final exam and that helped them out considerably. It brought them from, rather than having a 0 for 15% of the grade, they got a 93. In this case, now this is a part that I want you guys to bear with me. What's effectively this person, because their final exam replaced 2 grades, that means their final is worth effectively 45% of their grade. That's a little bit tricky, but it's kind of positive in some respects. Now, if we were to look at a slightly different situation, and let's say that somebody got a 0 on the final, they skipped it. Well, in this case, this is a little bit different perspective to have on it. Their final doesn't replace anything, but since they got a zero on one of their other exams, their final doesn't help them in that case. So you could kind of look at it as, well, their final ended up being worth 30% of their grade. Now, if somebody did really well going into the final, let's say they had a 90, let's say a 100, that would be not crossed out. And they did take or they skipped the final, well, their final is going to be worth 15% of their grade. Are they assured an A? Nope. Did they do really well? Yeah. Um, could they have probably come in and, just kind of blown through the final as quickly as possible and still ended up with an A? Probably. But if you don't take it, the, you 0 points. Okay. Those are all the things that I wanted to go over today. I feel like everyone's had opportunity to ask questions. If something comes up, please email me. I've got a couple of emails that I need to respond to. Um, but other than that, I want to say thank you guys very much for hanging out with me on Mondays and Wednesdays. I really appreciate it. You guys were awesome. I love that you engaged. I love that, attendance was always very positive. And I feel like we had a good experience. I hope that you learned a lot, and I hope that you do well in everything that you go forward with. So, um, thank you all very much for coming to class, and thank you. What that mean? It replaces the other grades, or um, the final potentially can replace grades. Thank you. Um, okay, folks. If I don't see you before Wednesday, or if I don't see you on Wednesday, best of luck for those of you graduating. Go get them. Um, and reach out to me if you got anything. Deal?